Okay, second part of section 18.1 on viruses, and now we're going to look specifically at animal viruses and start with this chart that was in the book that goes through how they classify viruses with animals, and the way they classify them, there's 6 different classifications based on their nucleic acid. So I'm going to kind of zoom in on this a little bit. Class 1, we've got our double-stranded DNA. Makes sense, okay, nothing special there. Class 2 here, we've got our single-stranded DNA, which is a little bit different, so that means that the DNA, we're going to have to make a complementary, or we're going to be able to use that DNA to make messenger RNA, and then we can go through the whole process of making the proteins that we need. So not a huge deal of a difference there with the DNA and single-stranded or double-stranded. We're not going to talk much about the double-stranded RNA. We're just going to kind of let it go, and the book spends a lot of time looking at the different types of single-stranded RNA, because if we have single-stranded RNA, we're going to have to go through some abnormal things within the cell to use that mRNA, or excuse me, to use that RNA from the virus effectively to make more viruses. Okay, so one of the simpler types here, type 4, has our single-stranded RNA, and once it enters the cell, it immediately acts as messenger RNA, and that kind of makes sense. Like, we've got RNA there, so let's just use it to make the proper proteins immediately. The thing that we have to figure out is, how do I use this messenger RNA to make, or excuse me, this RNA as it comes in here, to make more copies of itself, which there's specific enzymes that can do that. We're going to spend more time looking, excuse me here, at type 5. And the type 5 is a single-stranded RNA. Once it enters into the cell, that RNA is going to be used as a template to make new RNA. Okay, and so to discuss that, we're going to look at a diagram that's in the book, because this is a pretty special situation here. Okay, so here's a diagram of a type 4 virus, and a couple things here. This ends up being a single-stranded RNA that's inside of it. This is also the type of virus that has an envelope around it, so we've got our capsid in here. The protein that's surrounding the red is our DNA, okay, or excuse me, our single-stranded RNA, and then it's surrounded by a membrane. And so what we're going to do here is we're going to display what happens when we have a type 4 with a single-stranded RNA that's used as a template to make messenger RNA. We are also going to talk about how an envelope is made, okay, to go around this bacteria. So first of all, no details on this, but we're getting our virus inside of the host. Okay, so an animal cell here. Once it gets inside of the host, it breaks apart, and we have our viral single-stranded RNA is released. Okay, now something that they're not showing here is that inside of this virus, as well as having the single-stranded RNA, it's going to have some sort of an enzyme that its job is to take the RNA, the viral genome, the RNA, and basically use it to make messenger RNA copies of itself. Okay, so that it can, anyway, but that enzyme is going to be inside of here. We're going to see this when we look at the HIV virus on the next slide as well. Okay, so anyway, we have this viral genome, the single-stranded RNA. That's going to be used to make a template. Okay, it's going to be used to make, or as a template to make messenger RNA. Okay, so this, again, we're kind of using it as if it's DNA, but it is RNA, and so it has to be a new virus, because inside of a cell, animal cells don't normally have any kind of an enzyme that would take RNA and make a messenger RNA copy from it. Okay, so there has to be a viral enzyme that does that. Anyway, so we have this messenger RNA that can do what it does. We'll get back to that in a second. The other thing that happens is this RNA copy is going to be used as a template to make a complementary copy, which would be identical to the original genome. Okay, so if we kind of go through complements here, the messenger RNA ends up being complementary to the genome, which means we can use it as a template to make more genome, okay, for the virus. And so that's a step that's happening over here. We're going to make a bunch of copies of the viral genome. If I step back to the messenger RNA, there's going to be genetic code here to make all the capsid proteins so that we can surround our genome RNA with the protein, with the capsid. But then there's also going to be genetic sequence here to make glycoproteins. And so what's going to happen is this messenger RNA is going to attach to ribosomes that are on the rough ER, okay, and it's going to make glycoproteins in the rough ER. If we go back a bunch of chapters, this should sound familiar. Those glycoproteins, we're going to put them in a transport vesicle, which is going to transport them to the outer membrane. This vesicle will actually become part of the outer membrane, and then the glycoproteins that are on the inside of this will then end up on the outside of the cell membrane. So we have a bunch of glycoproteins on the outside of the cell membrane. When we have our assembled viruses with their viral genome in the middle and then the capsids on the outside, this is basically going to be exported here to the cell membrane, the host membrane, and it's going to get bound by the host membrane that has all these glycoproteins on it. Okay, so that's how we get our envelope. The purpose of those glycoproteins is they are the way that this virus, when it goes to a new cell, that's the way that this virus gets accepted by the new cell. They give a signal to the new cell that basically says, accept me, okay, or take me in, and that's how this will attack the new one. But so that's the idea of an envelope being made. It is also showing this type 5 classification of viruses, where our RNA is going to be used to make messenger RNA, and then there's going to be another process that makes more copies of the genome. So a couple things there. Next, we're going to look at the sixth type of, if I pull this back up, if I can, here we are. The sixth type of a virus, okay, uses the RNA for making DNA. Okay, and so this is something that's kind of special. Again, normally in a cell we go from DNA to making RNA. In this case, we're going to go from RNA to making DNA. Okay, and we see this, our example here is HIV. So our next slide, we're going to go ahead and take a look at HIV and how that works within a cell, and then kind of go from there. All right, here I have a picture of the HIV virus, actually a diagram of the HIV virus, and the reason I wanted to show this, on the previous diagram that you were looking at, we were talking about an organism, a virus that did a RNA to mRNA kind of transcription, and that's really abnormal. So I had mentioned kind of in passing that there had to be enzymes within the virus that did that. Well, the way HIV works, this is a type 6, where it takes single-stranded RNA, type 6 virus, single-stranded RNA, and it uses that to actually make DNA. Now, this is the reverse of a normal process. The normal process is we make RNA from DNA, so normally it goes this direction inside of a cell. So the cell doesn't have any machinery to take it from RNA to DNA. It doesn't have any machinery for that. So what they're showing in here is an enzyme called reverse transcriptase, right here, the green on each one. That reverse transcriptase, its job is to do the reverse of transcription, and transcription takes DNA to RNA. This takes RNA to DNA, and just to show that there is an enzyme, so no longer is it just we've got our capsid with some type of nucleic acid on the inside. We have our capsid with our nucleic acid on the inside, and now we also have some other enzymes, extra viral enzymes, that can do jobs that the hosts won't be able to do. Okay. Also, just to point out, this is a virus that has an envelope, glycoproteins on it, just like the previous one. We're actually going to look at the specifics here of how let me delete all these things, how HIV works, and then talk about medications that they've been trying to figure out to halt the HIV into AIDS transition. So anyway, there's a lot of similarities to this and the previous slide, so I'll be able to skip through some things. We have the entry of the HIV virus. Just the envelope is going to become part of the host cell's membrane, and we're going to dump the capsid in here and break it apart, and then we have our RNA. And then this is the big spot right here where there's a big difference. One of the big spots is that we are going to take the viral RNA and we're going to do reverse transcription okay, to end up with the complementary DNA to the viral RNA, reverse transcription, excuse me. And then we're actually going to kind of do 2 rounds of that so that we can get 2 complementary strands of DNA so that it'll be kind of like normal human DNA with 2 strands. Anyway, and then this is where things get a little bit tough for HIV, and one of the reasons it is such a challenge medically is it doesn't just take over a cell and destroy it or something like that. Instead, it takes a provirus form. It's kind of like the lysogenic cycle. So here we have our provirus, where our HIV DNA will actually insert itself into the host genome. Okay, so it's inserted itself into the host genome, and then it'll just kind of exist there. And as this cell is going about doing its daily activities, it will be taking some of the genes from the viral DNA and doing transcription, transcribing mRNA. Let me switch to the point here. Transcribing mRNA. Some of that mRNA is going to be used to make our capsid proteins, and some of that RNA is going to be used as RNA to go inside of a virus. So we're actually going to produce HIV viruses. Some of the mRNA is going to be used to code for the glycoproteins, just like we saw on the previous slide, and so you're going to end up with glycoproteins on the surface here. And then this virus is going to end up leaving the cell, and it can go and infect another cell. One of the reasons this is tough is it never really destroys this host cell. The host cell is able to continue to grow and do its job and infect other cells because we're releasing viruses. And so there's not a huge amount of change for this cell. It can be challenging. So medication-wise, the things that they're trying to do, they're taking advantage. One, they know this reverse transcription process that happens here. They have found some medications that can halt reverse transcription. And so that's one of the things that they try to do is get you some medications that can halt reverse transcription, because then your HIV won't be able to spread. Okay, and you'd have to continue to take them. Then the other options, other things they have, is down here where we assemble the actual virus. So we take all the proteins and join them together with the RNA. There's actually, this process requires some proteins to help it with the assembly process, and so they will. Excuse me, trying to check my time here. Okay. Anyway, there's some medications that can try to halt that assembly process, and so oftentimes people with HIV, they will have them take several medications to try to halt different parts of this whole process. Anyway, so that's how HIV works. Here we've covered a bunch of different ways that viruses can reproduce. So we had the lytic cycle, the lysogenic cycle. We showed one general form of an RNA to RNA virus, and here we have an RNA to DNA reverse transcription type model with a provirus. And anyway, so that's HIV. Last little bit, the book goes into the evolutionary thoughts here with viruses, and the thing I wanted to point out is, as far as evolutionary history goes, it would be real easy to look at viruses and say, if this is the beginning of time and this is currently, that viruses probably, they're so simple, that viruses probably popped up first, the simplest thing first, and then maybe prokaryotes were next, and then eukaryotes a little bit later, and whatever. And I think at some point scientists did believe that. Now that they're able to sequence the nucleic acids in viruses, what they're finding is that viruses are more closely related to their hosts, typically, than they are to other viruses, which would indicate the viruses haven't evolved from other viruses. Rather, they are probably segments of DNA that have somehow developed from their hosts and then mutated since then. So I guess the two big things here is, one, they don't think viruses were the first things to have evolved, even though they are very simple. And the other one is that they really don't have any good evidence to support any of their hypotheses of how viruses did come to be. It's just a very unknown area, and so there's a couple theories that are thrown in the book. They're logically credible, but as far as experimentally being able to do much with them, it's not quite there. So just to throw that your direction on the evolution, and I think that's it. So we will get to we're skipping section 18.2, and we'll get to section 18.3 here for you shortly. So.